Students, today we can see about the prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae or cyanobacteria, mycoplasma and PPLO. PPLO means pleuropneumonia-like organisms. These are organisms which can cause mild pneumonia, but it will not cause severe pneumonia, just like caused by the bacteria Streptococcus pneumoniae. Okay. Now these prokaryotic cells are generally smaller than that of the eukaryotic cells, but they can multiply very rapidly or faster than that of the eukaryotic cells. And these prokaryotic cells also vary in their shape as well as size. When we see the bacteria, there are four basic shapes of bacteria. They are cocci or spherical shaped bacteria, bacilli or rod shaped bacteria, spirilla or spiral shaped bacteria, and vibrio or comma shaped bacteria. So these are the four basic shapes of bacteria. When we see about these prokaryotic cells, they exhibit a wide variety of shapes and functions even though they are fundamentally similar. Okay, the prokaryotic cells are surrounded by a cell wall and a cell membrane. Okay, if you see the prokaryotic cells, it is surrounded by a cell wall, and inner to the cell wall a cell membrane or the plasma membrane is present. Okay, if you can see that in the picture also. What is present here? Cell wall. This is the cell wall which is present, and then a plasma membrane is also present in the prokaryotic cell except the mycoplasma. Okay, if you see the mycoplasma, they are not surrounded by any cell wall, but the prokaryotic cells are surrounded by a cell wall as well as a cell membrane. Okay, then inner to the cell membrane is a fluid matrix which is called the cytoplasm. Okay, so inner to the cell membrane is the fluid matrix called the cytoplasm, and they do not have a well-defined nucleus. If you see this picture here, you cannot see any nucleus. Can you see any nucleus here? No, but only the genetic material is present, isn't it? So they do not have a well-defined nucleus, and the genetic material is naked and not enveloped by a nuclear membrane. So nuclear membrane is absent. Then the region where the genetic material is present is said to be as the nuclear region or nucleoid. Okay, then it is having a genomic DNA in the single chromosome. Okay, so what you are seeing here is this is the single chromosome. Generally the prokaryotic cells will be having a single chromosome, and in that chromosome the genomic DNA will be present. Okay, why we say it as the genomic DNA? It controls all the characteristics of the that cell, that prokaryotic cell. That's why it is called the genomic DNA. It contains the genes that control all the characters. Okay, so a genomic DNA is present in the single chromosome. In addition to that, the bacteria also have a small circular DNA outside the genomic DNA. Okay, they have a small circular DNA outside the genomic DNA, and that is called the plasmid. What is it called? It is said to be the plasmid, and this plasmid will be conferring certain unique phenotypic characters to such bacteria which have the plasmid in it. Okay. So what phenotypic characters can be conferred by this plasmid? For example, we can say resistance to antibiotics. Okay, if the plasmid is there, and that plasmid, if it is having the gene for antibiotic resistance, then if we place the bacteria in a medium containing antibiotic, it will not be destroyed. It can resist, it can withstand that antibiotic due to the presence of that antibiotic resistant gene. Okay, so the plasmid can confer certain characters, certain phenotypic characters to the bacteria like resistance to antibiotics. Okay, now as I told, they do not have a nuclear membrane, and they do not have membrane bound organelles. Okay, membrane bound organelles are also absent in such prokaryotic cells. Okay. but they have ribosomes. Ribosomes are not surrounded by membrane, so they have ribosomes. Okay, then they are also having a specialized membranous characteristics, that is mesosome is present. Okay, it is a specialized differentiation of the cell membrane or the plasma membrane. What is it? Mesosome. Okay, this mesosome is nothing but the infolding of the plasma membrane. That is, the plasma membrane or the cell membrane will be folding towards the inner side of the cell, and that is said to be as mesosome. Okay. Now, when we see about the cell envelope and its modifications, in most of the prokaryotic cells, especially the bacterial cell, they are having a complex cell envelope. Okay, they are having a complex cell envelope. That cell envelope is formed of three layers. What are the three layers? They are glycocalyx, followed by that will be the cell wall, and then the plasma membrane. Okay. So the bacterial cell is having a complex cell envelope which is formed of a three-layered structure. What are the three layers? The outermost is the glycocalyx, followed by that will be the cell wall, and the inner one will be the cell membrane, that is the plasma membrane. Okay, and even though each layer of the envelope will be performing distinct function, different functions, they act together as a single protective unit. So the main function of the cell envelope or these three layers is what? Protective in function. Okay, and the bacteria are classified into two types. Okay, on the basis of the difference in the cell envelope as well as how they respond to the staining procedure developed by Gram. Okay, so based on the cell wall or cell envelope difference as well as how they stain, the stain which is developed by Gram. Okay, based on that the bacteria are classified into two types. They are Gram positive and Gram negative bacteria. So how the bacteria are classified? The bacteria are classified into two groups on the basis of the difference in the cell envelope and the manner in which they respond to the Gram stain. Gram stain is a stain which is developed by Christian Gram. Okay, it is somewhat a purple colored or violet colored stain. So whether they take up the stain or not, based on that the bacteria is classified into two. What are they? That is Gram positive and Gram negative bacteria. Okay. Now, if you see the Gram positive bacteria, they will take up the Gram stain. So, what is a Gram positive? Gram positive means they take up the Gram stain, and Gram negative means they do not take up the Gram stain. Okay. So, Gram positive means they take up the Gram stain means what? When you add the stain, they will take up the stain and they become colorful. Okay, see this is the Gram positive bacteria. Here what you are seeing, this one is the Gram positive bacteria, and you can see here this is the outer cell wall. Okay, this is the cell wall which is purple in color. It is due to the taking in of the Gram stain. So when they are taking the Gram stain, their cell becomes colorful and they are the Gram positive bacteria. Okay. On the other hand, if you see here, this is the Gram negative bacteria, and see they are having the outer envelope there which cannot take up the Gram stain. Okay. So a lipopolysaccharide is there, isn't it? Here you can see the lipopolysaccharide. Okay, lipopolysaccharide membrane is there, that will not be taking up the Gram stain, so they will not be colored. So here they are not colored, only inner layer is colorful, isn't it? But externally when we see, we cannot see the color. Such bacteria we can say it as the Gram negative bacteria. Okay. In the previous slide, we have seen that the cell envelope is formed of glycocalyx, cell wall and the cell membrane, isn't it? The glycocalyx will be differing in composition and thickness in different bacteria. It can be a loose sheet called the slime layer in some of the bacteria, and in some it become a thick and tough structure called the capsule. So here in the picture also you can see, okay, the first picture. So this is the slime layer, the green colored structure which you are able to see is the slime layer. Okay, it is the loose sheath. The glycocalyx forms a loose sheath called as slime layer, but in the second diagram you can see this is a very thick and tough layer, isn't it? And this is called what? The capsule. Okay, so this is about the glycocalyx. So it differs in different bacteria. Now when we see the cell wall determines the shape of the cell, and it provides a strong structural support to prevent the bacteria from bursting and collapsing. Okay, so the cell wall will determine the shape of the bacteria of the cell, and it provide a strong support to prevent the bacteria from bursting or collapsing. That is, if you keep a bacterial cell in a hypotonic solution. What is hypotonic solution? A solution which is having more water concentration. Okay, the concentration of water is more in the solution than that of the cell cytoplasm. Okay, now when you keep this bacterial cell in such a hypotonic solution, what will happen? The water will be moving from a region of high water concentration to a region of low water concentration. So the solution is having more water, and the cell cytoplasm is having less water. So from where to where the water moves, the water will be moving from the solution into the cell. So what happens to the cell? The cell will be swelling. When more and more water enters inside the cell will swell, but it will not burst. Why? Due to the presence of the strong cell wall. Okay, but on the other hand, if you are keeping an animal cell in a hypotonic solution, what happens? The animal cell is surrounded by a cell membrane. So when more water enters inside, it will swell and it will burst because they are not having a thicker or a strong cell wall. So there is chance for bursting of the cell. But here the cell wall will prevent the bacterial cell from bursting or collapsing. Okay, now when we see about the plasma membrane, the plasma membrane is semi-permeable, or we can say it is selectively permeable. What is meant by semi-permeable? It can allow only certain materials to pass through or to pass out. Okay, so it is said to be a selectively permeable membrane. So the plasma membrane is selectively permeable in nature. Then already I told about the mesosome, isn't it? So what is mesosome? It is formed by the extension of plasma membrane into the cell. That is, the plasma membrane will be folding inside the cell, towards the inside the cell, and that is called mesosome. And these extensions will be in the form of vesicles, tubules, and lamellae. Okay, so how it can be seen? It can be seen as vesicles. Vesicle means the membrane will form a circular manner, just a sac-like structure it will be formed. Okay, like this it can be formed. This is vesicles, or it can be tube-like, a small tube-like structures, that is tubulae, or membranous structure. Membrane means a single membrane like this. Okay, so the mesosomes can be in the form of vesicles, tubules, or as well as lamellae. And what is the function of this mesosome? They help in cell wall formation, DNA replication, and its distribution to the daughter cell, or it can also help in respiration and the secretion processes, and it increase the surface area of the plasma membrane, and enzymatic content can be increased. Okay, so these are the functions of this mesosome. Here you can see the mesosome. So these are the functions of the mesosome. Okay, so what are the functions? They help in cell wall formation, DNA replication and distribution to daughter cells, help in respiration, secretion, increase the surface area of plasma membrane and enzymatic content. Okay. Then in prokaryotic cells like cyanobacteria, in addition to this mesosome, they also have other membranous extension into the cytoplasm, which are called as chromatophores. What is it called? Other membranous extensions called chromatophores are also present where in some of the bacteria, since especially cyanobacteria. Okay, and that is called as what? Chromatophores. Okay, I hope you are able to follow this. Yes, so cyanobacteria has other membranous extensions called chromatophores. Now, what is the use of this chromatophores? They contain the pigments which gives color to that bacteria. See, chromo means color. Okay, so coloring agents are present in that. Coloring pigments are present in the chromatophores which give color to the bacteria. Okay, then when we see the bacteria can be motile or non-motile. Okay, some of the bacteria can move, some of them cannot move. Motile means they can move, non-motile means they are not capable of movement. If they are motile, they have thin filamentous extension from their cell wall called as flagella. So what is present? If they are motile, they have what? Flagella. Okay, they have thin filamentous extensions from the cell wall which is called the flagella. So in the picture also you can see the flagella. Okay, so this is the flagella. This here also you can see the flagella. Okay, now this flagella will be ranging in their number and arrangement. Okay, different bacteria will be having different number of flagella and their arrangement also varies. Okay, and the bacterial flagella is having three parts. Mainly three parts are there. What are the three parts? We can say three parts are there in this flagella. They are the filament, hook and the basal body. Okay, so what are the three parts? Filament, hook and the basal body are the three parts of the flagella. Okay, so the three parts of the flagella are a filament, hook and the basal body. So here you can see the basal body and this will be the hook region and this is the long filament. So these are the three parts of the flagella. Okay, the basal body is formed of protein structures which help in the growth of the microtubules. Actually, the filament is formed of microtubules. Microtubule is a tubular protein. It is nothing but tubular protein. So for the formation of the tubular protein, this basal body is helpful. Okay, then hook is the part which connects the basal body with the filament and it acts as a joint and it is flexible in nature so that it can help in the movement of the rigid filament. The filament will be somewhat rigid in nature. Okay, so for the movement of that rigid filament, this hook can be helpful. And the filament is the longest portion and it will be extending to the cell surface, from the cell surface to the outside. Okay, the filament is the longest portion and extends from the cell surface to the outside. So this flagella helps in movement of the bacteria. Okay, in addition to this flagella, the bacteria also have structures like pili and fimbriae. Okay, they are also having structures called what? Pili as well as fimbriae. The villi are made up of special proteins. Okay, especially they can help in primitive mode of reproduction. Okay, primitive mode of reproduction means conjugation. So in one of the previous chapters we have studied that. So during conjugation, two bacteria will come together and they will be exchanging their genetic material, isn't it? At that time they will be attaching with this pili. Okay, then in addition to that they are also having other structures called fimbriae. Fimbriae means they are bristle like fibers which are sprouting from the cell, which arises from the cell. Okay, in this picture you can see the fimbriae here, the minute hair like structures which are present. That is what the fimbriae. Okay, So these are bristle-like structures. Bristle-like structures means what? See, in a brush you can see, here in the picture you can see that, okay, small hair-like structures or bristle-like structures. Okay, so in the brush you can see that bristles, isn't it? The toothbrush you can see that like that. Here also, surrounding the bacteria we can see small bristle-like fibers. So these are called as fimbriae. And what is the use of this fimbriae? They help the bacteria to attach to any substratum like rocks in the streams or in any host tissues. They can attach to any substratum. It helps to attach. So that is the function of fimbriae. Okay. Ribosomes are present in prokaryotic cells. The ribosomes which are present in prokaryotic cells are associated with the plasma membrane of the cell. Okay, they are attached to the plasma membrane of the cell, and they are about 15 nanometer by 20 nanometer in its size. And generally they are made up of two subunits. The ribosomes are made up of two subunits. What are they? 50S and 30S subunits. Okay, the two subunits which are present are the 50S and the 30S. See here, you can see in this picture, you can see the two subunits present in them. That is, one is smaller in size and another is the larger in size. Okay, so the larger one is the 50S subunit and the smaller one is the 30S subunit. Okay, and when they are present together, they form the 70S ribosome. So don't think when we add 50 and 30 we will get 70. It's not like that. Okay, the two subunits are 50S and 30S. Okay, when they join together they form the 70S. Now what do you mean by that S in this? It means Swedberg's unit. Okay, the unit name is what? Swedberg's unit. It determines the sedimentation coefficient. Actually it is the measurement of sedimentation coefficient. Indirectly it measures the density and size of this ribosome. Okay, that's why we represent it as yes, it is Swedberg's unit. Okay, so the ribosome which is present in prokaryotic cell is 70S ribosome, remember, and in eukaryotic cell it is 80S ribosome. Okay, and what is the function of these ribosomes? The ribosomes are the site of protein synthesis. They help in the synthesis of protein. Okay, so at the time of that protein synthesis, several ribosomes can attach to a single mRNA. So here in the picture you can see a line. Okay, this line actually is the mRNA. Okay, and in that mRNA what is attached? The ribosomes are attached. What is mRNA? mRNA is a messenger RNA. RNA, you know, ribonucleic acid. It is another genetic material. So here it is mRNA means messenger RNA, which will be carrying the information for protein synthesis. Okay, so the ribosomes will attach to the mRNA. Many ribosomes will attach and they form the polyribosome or polysome. Poly means many. Many ribosomes will be attaching to mRNA and form the polyribosome or the polysome. Okay, so this polysome helps in the translation process. What is translation? Translation means synthesis of protein from RNA. Synthesis of protein from RNA is called translation. So the polysome helps in the translation process. That is, mRNA, from mRNA a protein can be synthesized. Okay. Then next is the inclusion bodies. Here we can see the inclusion bodies. The reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Okay, so all the reserve materials which are stored in the cell actually forms the inclusion bodies, and they are not surrounded by any membrane. So they will be lying free in the cytoplasm. Okay, for example, we can say phosphate granules, cyanophycean granules, glycogen granules. These are all the inclusion bodies which are seen in the prokaryotic cells. Okay, see, if you see in this picture also you can see that the here small round structures are there. Okay, smaller round structures. Can you see this? So these are some of the inclusion bodies which are present in the prokaryotic cells. In addition to that, some gas vacuoles are also found in blue, green and purple and green photosynthetic bacteria. Okay, gas vacuoles which are filled with gases can be found in the blue-green bacteria, that is the cyanobacteria, or the purple and green photosynthetic bacteria. Okay, so this is about the prokaryotic cell.